In this video, we're going to begin to understand the genetics of cancer, and we're going to try to figure out why cancer is often a disease of old age. So let's first look at this graph. You will see that in young people, so 5 to 14-year-olds, the incidence of cancer is very low. So 0.2% of the population in the world in that age group has cancer. So it's a very small percent. However, by the time people reach 70 years older, 6% of the population has cancer. Okay, and those are all cancers. So cancers of many different types, whether it's lung cancer, colorectal cancer, breast cancer, things like that. So, and that is a diagnosis, not necessarily a death rate, but we are seeing definitely this exponential increase in cancer over time. So let's try to figure out why that occurs. A key idea for this video is that cancer is a genetic disorder that comes from the accumulation of mutations in the genes that regulate the cell cycle. The biggest risk factor for developing cancer is aging. Over time, we accumulate mutations that are due to random replication errors or by DNA damage from carcinogens. And we'll learn more about both those things later on. But this picture basically shows how mutations can lead to cancer. Each of these arrows is basically representing a new change to the cell. So here is our initial cell, okay? And this first mutation is going to inactivate a negative cell cycle regulator. Okay, so that's something that would normally regulate the cell cycle by shutting it down. Okay, so something that stops the cell cycle at, say, a checkpoint. Okay, there's a mutation there and it shuts it down. Okay, then the cell develops another mutation. In this case, it's a positive cell cycle regulator and it's overacting. So we've learned that cells need signals in order to divide. So a positive cell regulator is kind of like one of those signals. Basically, the cell is being overreactive to signals and it's dividing too much as a result. Okay? These are things like growth factors, growth hormones, cyclins, all these things that help push the cell through the cell cycle. So far, we've shut down some of the checkpoints. We have sped up the cell cycle, but understand the cell cycle, right? Okay, it's got 4 parts, G1, S, G2, and M. There are multiple checkpoints that occur throughout this cell cycle to make sure that everything goes correctly. So just one negative cell cycle regulator and one positive cell cycle regulator being altered isn't enough to make the cell truly cancerous. It's starting to divide uncontrollably, but it's not really out of whack to form a tumor yet. Now we're going to have a third one. In this case, it says it inactivates a genome stability factor. So that might be something down here with the M phase, right? When at that checkpoint, normally they check to make sure that the spindle is attached to the centromeres of the chromosomes so they separate properly. Well, maybe that checkpoint doesn't happen, and so the chromosomes aren't attached to the spindle, they don't separate properly, and then you end up with cells with too much DNA or not enough DNA. That's genome stability. So now we're getting extra or missing chromosomes. That's a problem. And then you're just going to have to start to accumulate lots more mutations. So now that things are really going out of whack and that the genetic material isn't quite organized the way it should, and we're starting to accumulate these mutations, more and more mutations are happening rapidly, and you're going to get a cancer cell. So this is an important idea we're going to talk about a lot. You'll hear this. So typically 6 to 10 genes will need to be altered in order to make the cell cycle really go out of whack and the cell to become cancerous. Now, which 6 to 10 genes are going to be altered will vary from each patient, So let's explore a little bit more about what these genes might be that might regulate. So there are 2 main types of genes that regulate the cell cycle. The first one are those positive regulators. They are proto-oncogenes. Now, normally, these normal genes, unmutated genes, stimulate the cell cycle, okay? And they trigger cell division by producing growth factors, hormones, or other proteins that carry messages in the cell. So if we look at this picture, here's a growth factor, okay? Growth factors are made by proto-oncogenes. The receptors that receive the growth factor, they are also made by proto-oncogenes. These signaling molecules that basically tell the DNA what growth factor has been received, because there's this kind of chain reaction, those are all made by proto-oncogenes. So basically all of the proteins from receiving a signal that it's time to divide to turning on the DNA that then starts the cell cycle, all those proteins are made by proto-oncogenes. They're telling the cell it's time to go, okay? And those are important because if there's DNA damage or the body needs to grow, the cell needs to know it's time to divide. So think of proto-oncogenes like a gas pedal for the car. They make the car go. The problem is when mutations occur in proto-oncogenes, they produce oncogenes. So it's a little weird. They changed the name. Onco is referring to cancer. So these are cancer genes. And they accelerate the cell cycle. So it's not just enough to make it go. They make it go too fast. So if you had the analogy of the car, think of an oncogene as a stuck gas pedal, okay? The proto-oncogene was overstimulated. It's working too well, okay? The gas pedal's stuck. It's going too fast. And so you can see here that in this picture we saw before, if we go back, that you needed a growth factor to bind, and then it started a signal transduction pathway. Well, here with an oncogene, look, there's no growth factor, but the signal transduction pathway is happening on its own. So basically, the cell is dividing even when the neighbors aren't telling it it's time to divide. So these proteins are being made at the wrong time in the wrong amount, and they're telling the cell cycle it's time to go, even though the body doesn't want the cell to divide. The next type of gene that regulates the cell cycle is a tumor suppressor gene. And this gene does exactly what its name says. It suppresses tumors. So these are negative cell regulators. They shut down the cell cycle. They suppress the tumors. So their job normally is to protect the cell from developing cancer by regulating cell checkpoints. Think of them like brakes to the car. Okay, the cell's going to stop at those 3 checkpoints, the G1, the G2, and the M checkpoint, and make sure everything is okay. Now, how exactly do they stop it at the checkpoint? Well, they do the things that the checkpoints check for. So we talked about DNA repair being at many of the checkpoints. We've talked about things like checking for the spindle being connected, checking that DNA replication occurs. Okay, that's the job of tumor suppressor genes. They're those checkers at the checkpoints to make sure that everything has occurred properly. Okay, so if they can't fix the problem, then they're going to trigger apoptosis and they're going to kill the cell if too much damage occurs. So our mascot for the class is P53. P53 is our tumor suppressor gene, okay? And here you can see this is a normal cell. Now it has extensive cell DNA damage in this cell, and so P53 proteins are going to be made by this gene, and those proteins will eventually lead to apoptosis, basically the digestion of the cell from the inside out, okay? And that's a good thing. It's going to stop the cancer. It's going to stop tumors from forming. If the genes are mutated, they don't make the proteins that stop the cell cycle. It's like cutting the brakes on a car. So that's an important idea. Okay, so the cell basically can't stop. It keeps going through checkpoints despite harmful mutations. Okay, So it's important to note that tumor suppressor genes are inactivated in cancer, whereas proto-oncogenes are overactive in cancer. Okay, so here we can kind of see this relationship. Okay, so an oncogene, which is a mutated proto-oncogene, it promotes cancer. Its expression in normal cells is low, right? We don't want to form cancer in a normal cell. But when we get to cancer, we have high expression. That's the stuck gas pedal, okay? So it's got too much gas. It's making it go too fast. A tumor suppressor gene prevents cancer. They are very active in normal cells, but in cancer cells, their activity is low. Think of them like no brakes. They cut the cell cycle. Both of those things need to be in place, okay, for cells to go awry. You need to not only make the car go, right? Pushing down on the gas is great, but if you have a brake, you can still stop, okay, and prevent, in this case, the car from going or the cell from dividing and cancer from occurring. But if you have the gas stuck and then you don't have brakes, that's going to cause the problem. So normally we have these checks and balances in our cells. The need to go is balanced with the need to stop. Okay, cancer cells, we shut down the brakes, okay, and we make the cell go too much. So the cell cycle is tightly regulated by genes that activate or inactivate the cell as the cell progresses through the checkpoints. On average, 6 to 10 of these genes must be mutated in order to inactivate the cell cycle controls and for the cell to become cancerous. So like I just mentioned, there's this balance between tumor suppressor genes and proto-oncogenes, and that's going to result in normal cell growth. Okay? But in malignant cell division, we see that they've lost this balance. There's less function of tumor suppressors. There's a gain of function in the oncogenes, and that's going to cause the formation of tumors. So let's talk a little bit about what types of mutations can occur. So mutations can kind of come about in two different ways. They can either be somatic or germline. Somatic mutations are mutations in body cells. They are not passed on to the offspring because offspring are made through sperm and egg, not our body cells, right? We don't pass our skin to our kids, okay? So every time a cell divides, it must replicate its DNA, and mistakes are made about every 10 billion base pairs. So despite everything we've learned with DNA replication, sometimes errors do happen and they get missed, and mutations happen naturally. There's nothing you can do about that. What you can prevent are environmental conditions, things like UV damage or chemicals in cigarettes. They can damage DNA and they cause additional mutations. Germline mutations, on the other hand, are found in eggs and sperm. We call those our germ cells. Those are cells in the ovaries and testes that divide by a special type of cell division called meiosis to make eggs and sperm. Because they're in the hereditary cells, these mutations are inherited and are passed on to the offspring. When cancer runs in families, it's due to these mutations. So let's take a look. We will learn more about pedigrees in this unit, but a pedigree is essentially a family tree. Squares represent men, circles represent women, and then colored in circles or squares would represent individuals in the family who have developed some sort of trait. In this case, it's going to be individuals who have had either breast or ovarian cancer. So in this family, we are looking for mutations in the BRCA1 or 2 genes, and they increase the risk of developing breast cancer. Okay, they are a tumor suppressor gene, and sometimes people inherit mutated copies of these. And so basically what it means is they're one mutation closer to developing cancer. So here you can see in this family, we see a lot of individuals who are young. So females who have had breast cancer or ovarian cancer well before the age of 50. And that's usually the indication that there might be an inherited germline mutation in the family. So this picture is just another review what the significance of these. So if you inherit a germline mutation, like we can see, so this red sperm is supposed to represent the mutated DNA, okay? So the father's passing on the mutation in this case. The embryo that results, every single cell in that embryo is going to have that mutation because the very first cell received the mutation from the sperm. Which means as that embryo grows, the entire person carries that mutation. Every cell in the body is going to carry that mutation. And so therefore, every cell is now at a slightly increased risk if that mutation is cancer causing. Then when that person goes to make their own gametes, in this case if it's a man, their own sperm, there's a chance that he could pass on that mutation to his offspring. We'll learn more about why half the gametes carry the mutation in future units. Now, a somatic mutation, we can see that the gametes are perfectly healthy. There are no mutations in cancer-causing genes. However, at some point, the embryo develops a somatic mutation. So perhaps, notice it's just in a little localized area. It's just in a small, probably one cell, and then as that one cell divides, it's going to then pass it on to its progeny, the cells that result. So that somatic mutation in the embryo may have been due to an environmental factor. Perhaps the mother that was carrying this embryo was exposed to a chemical, and that chemical triggered it, or maybe there was just a mistake in DNA replication. So this somatic mutation now is present in this embryo, and as it divides by mitosis, it'll pass it on. But there's only going to be a small area that's affected by that mutation, okay? Somatic mutations are kind of like localized body cell mutations. Not every cell in the body is going to have it because it wasn't inherited. As a result, look, this case, the cancer is maybe up in the shoulder. So maybe it's a skin cancer or a bone cancer up in the upper arm, things like that. But that tissue isn't passed on to the offspring. So none of the gametes have it. Okay? So it's not going to be passed on. It was a sporadic cancer. Okay? Which we will talk about in a little bit. So let's talk about this idea of two-hit hypothesis. Scientists like to refer to mutations as hits, okay? So what this means, this two-hit hypothesis means, is that mutations in both the copies of a gene, one from each parent, are usually, I don't say always, but usually required to turn off a gene. And that's going to take you one step closer to developing cancer. So it's important to remember that as humans, we are something called diploid. And what that means is we have two copies of every gene. One chromosome came from the mom, and one chromosome came from the dad in the sperm. So we have two copies of every gene. Okay? Now what the two-hit hypothesis says basically is in order to knock out a gene, in order to fully make it a cancerous gene, you got to take out both of these copies. So it's not just a single mutation in a gene, it's actually usually two mutations to result in an ineffective cancer or cell cycle regulating gene. So in this case, somatic mutations, you can see that this person here was born with no mutations. As time goes on, they get a somatic mutation, and then as time goes on, they get another somatic mutation, and now they have one of 6 to 10 genes that are mutated, that are needed to cause cancer. So that basically means that they are on their way to potentially developing cancer. Are they guaranteed? Not necessarily, but they're one step closer. Okay, now if someone inherits a mutation, one of their copies is already basically shut down. But if they get a mutation in the other copy, now they have one of the 6 to 10 genes mutated. And notice, if this is time, this happens a lot faster. So if you inherit a cancer-causing mutation, you're not necessarily guaranteed to get cancer, right? Because you got to get 6 to 10 genes mutated. But you're one step closer. You're one gene closer, basically, one mutation closer to developing cancer. And as a result, you might develop cancer earlier on. And so when we see people who develop cancer at a young age, whether it's childhood cancer or in their 20s or 30s or 40s, okay, those individuals, not always, but sometimes may have this inherited mutation. Okay, they've already inherited a mutation, so they have fewer mutations that they have to develop randomly through somatic mutations in order to develop cancer. So let's talk a little bit about what this means. So scientists like to divide cancers kind of into two groups. You either have inherited cancer or sporadic cancer. Okay, so inherited cancer is when an inherited germline mutation contributes to the development of cancer. You still need additional somatic mutations, right? Because you need 6 to 10 genes to be altered, okay? But you are one step closer. So here you can see inherited cancer right here in this pink box. We often see an early onset, like I mentioned before, because you're one step closer, you develop cancer faster. Okay, we often see it in multiple generations within the family. We might see, they say bilateral cancer, so that might be multiple people within the same generation who have cancer. And it might be a clustering of certain cancers. So certain genes tend to cause certain cancers more often. So like the BRCA1 and 2 genes, they tend to increase your risk in breast cancers, ovarian cancers, and prostate cancers. So we would see that if a mutation in the BRCA1 or 2 genes is inherited in the family, we would see those cancers happening more often within the family. So remember, inherited cancer doesn't mean you're born with cancer. It just means you're born with one mutation out of the many mutations that you need to develop for cancer. You're not guaranteed, you're just one step closer. Sporadic cancer, on the other hand, is caused by the accumulation of somatic mutations throughout one's life. Okay? So this could be by chance, just bad luck, mistakes in DNA replication happen, they accumulate, and cancer develops over time. Or it may be accelerated because you participate in risky behaviors. You might not eat well, you might not exercise, you might smoke, you might be exposed to chemicals such as air pollution or asbestos and things like that. And therefore, mutations happen a little bit faster, okay? But basically, you're accumulating these mutations in your body cells over time, whether it's by chance or by environmental exposures. So with sporadic cancer, we tend to see it later in life. It says 60s and 70s. Typically, we say over the age of 50 is when sporadic cancers start to be more noticeable and more common. There's little or no family history because it's kind of a sporadic, unique to that individual. Okay, and we don't see large clusters of it within the family. Okay, so just remember, somatic mutations are required no matter what for cancer. But if you only have somatic mutations, then you have sporadic cancer. It's kind of by chance, it develops on its own. Whereas if you have an inherited germline mutation, plus some other somatic mutations, then you have inherited cancer. So this just kind of shows you the vast majority of cancers are sporadic cancers. 70 to 85% of cancers, they happen by chance because of mutations, either because of DNA replication or environmental factors. Okay, and then 5 to 10% is heredity cancer. So that's clustering in the family because of an inherited gene mutation. So they can trace it back to a specific gene mutation that's inherited, like BRCA1 or a mutation in P53, things like that. Now, this last group, this 10 to 20% of familial cancer, is a little bit trickier because when we see cancer in a family, sometimes we can pinpoint it on a gene. Other times, it might not be due to a gene. It might be due to shared factors. Things like everyone in that family is exposed to the same environment. Maybe that family lives near a factory, and that factory is releasing cancer-causing chemical into the water. And so we're seeing this family develop cancer, and that's not because of an inherited mutation, but rather because of a shared environmental factor that's triggering more somatic mutations. So we can see that too. But it's not just a chemical in the environment. It might be that they have similar lifestyles, so things like poor diet, smoking, things like that. So in conclusion, 6 to 10 mutations, mutations in 6 to 10 genes, including multiple tumor suppressor and proto-oncogenes, are needed to cause the cell cycle to go out of control. So we have to shut them down. 6 to 10 genes need to be shut down. For each of those genes, often 2 hits, 1 mutation per chromosome in a pair, are needed to inactivate or overstimulate the gene. So really, when we say cancer is the accumulation of mutations, it's not just 6 to 10 genes. It's often 12 to 20 mutations that have to accumulate because you need 2 hits per gene. So that's a lot of mutations. And so it often takes a long period of time for enough mutations to occur and overcome the cell's regulatory mechanisms. That long time, that is why cancer is typically a disease of old age.